Okay, guys. So we've seen how the flow of these electrons, right, through the electron transport chain results in these energized mitochondria, right? Um, and now what we need to do is connect up that idea, the buildup of that electrochemical gradient, so-called proton motive force, um, to the synthesis of ATP. Now, this whole idea, this whole concept, is of course known as oxidative phosphorylation, right? Or the idea that the oxidation of ultimately the carbohydrates or amino acid backbones, whatever, their oxidation leading to the reduction of NAD plus and FAD to NAD H and FADH2, and then their ultimate oxidation and production of water. That oxidative cascade is directly connected to the formation of that phosphodiester bond, where we take ADP and make ATP. The basic idea that this is what was going on, that these oxidation cascades were directly connected or powering the formation of ATP. That had basically that idea had been worked out by about the 1940s, right? People were pretty, the evidence was just overwhelming that that oxidation sequence in mitochondria is what was resulting in the production of ATP. And people even knew that this proton motive gradient was getting created. Um, but because of all the decades before, since the work starting the 20s through the 30s of Emden, Meyerhoff, and Warburg, all the work that was done by the guys who had worked on TCA cycle, including Krebs, people were just sure that this phosphorylation event that was the result, the product of oxidative phosphorylation, was a result of some energized compound, that it was a substrate level phosphorylation event. And this compound even got a name in the search. It's compound Z. And as a result of the work that was done, like I said, up into the 40s, there was a multi-decade long search looking for compound Z, looking for that special high energy compound that was going to be made at high levels and was going to directly create ATP from ADP. There were entire careers built around this. There were reports periodically that compound Z had been identified, and then it wouldn't ever quite hold up, right? But people were just sure that's how this had to work, because that was the precedent in these metabolic energy creating pathways of glycolysis and the substrate level phosphorylation seen at TCA cycle. People were just sure that's how it had to work, but it never seemed to be found. It was like always just out of reach. Then in 1961, a famous chemist by the name of Peter Mitchell, who'd been working on this problem for a while, decided that this compound Z thing just couldn't be right. And he had a different idea. And Mitchell proposed that ATP was being synthesized as a result of direct coupling to the proton motive force, right? That something about protons moving through that electrochemical gradient, that was the power directly for making ATP. And this got a name, right? It's known as the chemiosmotic hypothesis. Right? And in sort of graphical form, it looks, something like this, that we've got this oxidative cascade through the electron transport process that we've been talking about, kind of idealized here, and just like one thingy, making water and pumping these protons out. We get this energized mitochondria. And then there is a system of some kind, Mitchell postulated, that directly couples the inflow of protons from that stored energy, from that electrochemical gradient, to power formation of ATP, right? This idea in 1961-62 was not well received. Mitchell got massive pushback, and you can imagine why, right? You have people, famous people, who've invested decades in searching for compound Z. This was the sort of radical break with the paradigm that just does not sociologically go over very well. I mean, there are people convinced if they could find compound Z, there's a Nobel Prize there. And Mitchell came in and said, no, you got it all wrong. So the hostility to this idea persisted for decades, even to the point from 1960s on as more and more and more evidence accumulated that, yeah, this is probably how this thing works. Even when I was first being taught this in the 1980s, some of the older biochemists who have been marinating in this for decades still referred to this, not exactly derisively, but as an hypothesis, and presented the compound Z as a fully valid alternative hypothesis. It's not established yet, right? The election's not over yet, right? Well, Peter Mitchell was right, like massively correct, right? Compound Z does not exist. The idea that Mitchell proposed was dead on correct, and he won the Nobel Prize for this idea. And the chemiosmotic hypothesis became the chemiosmotic mechanism, right? And again, that basic idea is, again, that we have some sort of system that's directly coupling the inflow of protons to the synthesis of ATP. And this came to be known as complex 5, right? If we had complexes 1 through 4 that gave us the electron transport chain, and it got another name because of the way it was originally identified. It's also known as the F1, F0 ATPase, right? And we're going to look at it in a little more detail here in a minute. But like I said, a whole lot of evidence accumulated demonstrating that this was the mechanism. This is how this worked. But proving this against the skeptics, that can be a challenge. How do you prove that something doesn't exist, right? That can be a tall order. So you've got to do some really convincing experiments. And one of them was say, well, okay, what if I build an artificial system where there's no metabolism going on, there's nothing. I can basically 100% confident that compound Z can't possibly exist. And if I can show nothing more than an electrochemical gradient, proton gradient, will synthesize ATP, that's a pretty good proof, right? And so one of the experiments that really just nailed this to the wall was done in exactly that way. And it involved doing something like this. So there are certain bacteria that have a protein known as bacteriorhodopsin. Bacteriorhodopsin is a membrane-spanning protein. It's all alpha helical, and in the middle of it is this extended chromophore. It's going to absorb light, called a retinal. These bacteriorhodopsins are direct orthologs of the pigments in your eye that allow you to see light, right? They're responding very much the same way, and they are photon-driven, light-driven proton pumps. So when the retinal absorbs light, it changes conformation, which kinks a helix and repositions some residues, shifts their pKs in this sort of tight little channel in the inside of the rhodopsin molecule, which results in a light-driven directional movement of protons from one side of the membrane to the other side of the membrane, right? So you can use nothing more than light to drive protons across a membrane. All right, so here was the clever experiment. It's possible to isolate pieces of this bacterial membrane, utter and completely pure, but intact, chunks of it, because the membrane is kind of self-organizing, I guess is a way to put it, right? So you can take these bacteria and break up their membrane that has this bacteriorhodopsin in it and get little patches of that, right, that have lipid and the embedded bacteriorhodopsin in it, and then incorporate those into artificial liposomes of defined composition, along with that complex 5 from, I think it was pig heart muscle where this was first done. Here's the idea. So there's nothing in the system other than if you do it the right way and you get the complex 5 oriented the right way, you add ADP and inorganic phosphate on the outside of these liposomes, and you let them sit there. And then it's like nothing happens. Then you turn on the light. The rhodopsin does its thing, right? And again, this is oriented, okay, if you do it the right way, you can get most of these patches oriented so that the proton flow goes into the liposome. And if the complex 5 is oriented in the right direction, now as this proton gradient builds up on the inside of the liposome, these protons in principle can flow through this complex. The prediction is now you have a light-driven ATP synthesis machinery. by doing nothing more than shining light, this system will make ATP. And guess what? That works, right? Works beautifully, right? So this is, again, this is one of those just beautifully elegant experiments, stripped down, basic set of components, goes right to the heart of a key prediction of this model and nails it, right? So this is one of the reasons we're pretty sure, or more than pretty sure, we're very sure, this is how ATP synthesis works when you have a proton gradient across the inner mitochondrial membrane. Okay, so let's talk a little bit more about this complex 5, right? As I said, when this thing was first identified, it was first identified as an ATPase, and for historical reasons I don't really want to get into at the moment, it came to be known as the F1F0 ATPase. More recent parlance, people have started calling it complex 5 because it's sort of the last complex in the oxidative phosphorylation chain in all organisms that use aerobic metabolism like this, right? And it's built in a particular way, and it's a really important way. So this is where all these ideas of membrane biology and protein structure and protein stability and all this stuff sort of all come together into this micro or this nano machine that is a sort of an electrically driven, electrochemically driven motor that's going to make ATP. So the F0 part of this machine is the piece that's embedded in the membrane, right? And it consists of about 150 kilodalton complex of 3 different subunit types. One is the so-called C subunit. It's basically, each C subunit is essentially a pair of a couple of helices that form a helical pair. And then there are 10 to 14 of those arranged in a ring that's then embedded in the inner mitochondrial membrane. Okay, off to one side, just one side of that ring is the so-called A subunit, and these make close contact. The C ring, the so-called C ring, some people will call it, and the A subunit make close contact. And then there are this sort of extended B subunits that interact with the A subunit of the F0 and rise up into the matrix of the luminal side of the inner mitochondrial membrane. Okay, that's the F0. Okay, now the F1 is this sort of large ball-shaped structure. It's about 400 kilodaltons in mass, and it contains the active sites. This is where the business of making ATP happens, is in the F1 components, right? And this is constructed of a series of different subunits. The business end are these alpha beta subunits, and they exist basically as a heterodimer, an alpha beta heterodimer, and there are 3 of those that then make the sort of business part, the sort of active site chunk of the F1, right? So we have a heterodimer arranged in sort of a trimer of heterodimers, right, that make the main part of this ball. There's a so-called delta subunit that connects to one of the beta subunits, and it bridges the F0 and the F1 because it makes contact both with one of the beta subunits and also with those extended B subunits that rise up from the F0. Now, the other key part of this machine is this sort of piece right in the middle, the so-called gamma epsilon stalk, right? So there's this pair of subunits, one of which is quite extended, extends all the way up into the alpha beta ball of the F1, and then another piece, the epsilon group, that's down at the base. And the gamma epsilon stalk is making very tight, close contact with the C ring of the F0, right? Now, that was sort of, we had to gross structure that for, going back some number of decades. But somewhat more recently, 10 years ago or something like that, the atomic structure of the F1, F0, and I think this was bacterial, but I can't remember, was solved in atomic detail. Not totally, but almost all of it, right? So here's another look at it. Here again is that F0. There's those C subunit rings, right? Here's the pair, here's another pair, right? Forming that sort of collar-like structure that's embedded in the bacterial membrane. There's the A subunit. Off to one side, here's that extended B subunit coming up, making contact through the delta subunit up here with the F1, right? And here's an alpha, a beta dimer, and we got 2 more heterodimers are on the other side, and there's that gamma epsilon stalk right in the middle. Okay, now what some of you may remember, may not remember, is the key aspect, right? Big picture of what's going to happen here is that proton motive force, the flow of protons, is basically going to drive rotary motion of that F0 ring within the plane of the bilayer. It's going to spin, right? And the gamma epsilon stalk, base of the gamma epsilon stalk are stuck to that very tightly to that F0 ring. So when the F0 ring spins, the gamma epsilon stalk is going to also spin. This is sometimes known as the rotor of the complex 5, the F0 ring and the gamma epsilon stalk. Some people also will call the gamma epsilon stalk the axle of the motor, right? Now, for this thing to work, what has to happen is this axle has to turn within that ball, right? And we're going to see why here in a little bit. But that means the F1, the trimer of heterodimers, it has to resist the torque generated when the axle spins, right? And that's what the B subunit is for. It comes over here, makes contact with one of the B subunits, goes back down, makes contact with this restrained complex of the C subunit in the bilayer, so that as the gamma epsilon stalk spins, right, the alpha beta F1 ball doesn't. It stays still, and the gamma epsilon stalk spins inside of it, right? Now, there's a key thing here. It's going to become really important. For this synthetic machinery to work, right, this mechanical movement has to establish asymmetries within what is otherwise this symmetric ball of alpha beta heterodimers in the F1A part of the machine. That asymmetry comes about as a result of an asymmetric shape of that gamma epsilon stalk, that axle, right? And so as it moves, that asymmetry is going to sweep around within that ball, and that's key to how this machinery is going to work. Okay, so that is the machine, and in the next video, we'll take a look at how that movement synthesizes ATP and how that is ultimately then coupled to the flow of protons across the intermitochondrial membrane.